Hello my dear students, I hope all of you are doing very well and all of you are listening our all the lectures that we have taken since the first day of our this series of quality assurance. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute at Greater Noida. So, we are continuing the learning of unit 3 of quality assurance in B Pharmacy 6th semester, and this is the 20th lecture and also the last lecture of unit 3. So, today I am going to tell you about lots of things in which cases we will facing some issues and our study might be recalled or it might be disqualified. So, today I am going to tell you about the reporting of the non-clinical laboratory results. If you do some non-clinical laboratory studies, how you will report, what will be the necessary things should be available in your report. Then you will also have to tell I will also tell you about the storage and retrieval of the recording data, retention of the records, disqualification of the testing facilities, ground for disqualification, on which cases your study might be get disqualified, and notice and opportunity for hearing on proposed disqualification, final order on disqualification. If your study get disqualified, then what will be the final order? These things you are going to learn. Let me start with the reporting of non-clinical laboratory study results. So, obviously, a final report shall prepared for non-clinical laboratory study. It shall include, but not necessarily limited to the following. It means, following should be there. First, the name and address of the facility performing the study and the dates on which the study was initiated and completed. This is the main primary and basic things should be included in your study. Name and address of the facilities performing the study, where you are actually working, where your entire that is a clinical work actually working, you have to mention including the date, because by using the date the health officials or health inspector will be able to understand when it was actually began and what are actually going on. Objective and procedures of the stated in the approved approval protocol, including the any changes in the original protocol. Objectives and procedure. Why objective and procedure important? Because you have to understand that if you start your work, there might be some objectives, like if you are working on some anti-cancer project. So, obviously, your objective will be showing that yes, your drug is showing some anti-cancer activities. Your drug is showing some anti-cancer activity. This will be your objective to that is a show. And procedure, how you are actually concluding that yes, my drug having some anti-cancer activity. Also, the statistical methods employed for the analyzing the data. Many of the cases it was found that for differential design we are focusing or we are actually following different that is a statistical models or statistical methods, but after conduction of the studies, whatever the sufficient result was supposed to get, it was not achieved, but it is showing some result which is much more beneficial for another kinds of therapeutic activity. Okay. And recently nowadays this is the era of machine learning, SLR, multiple linear regression that I already have mentioned in my previous lecture, deep learning which is with the your that means this thing called neural network. Okay. So, this is the era of machine learning. So, if you properly mention your statistical methods and if you give the data with the data scientist, and if you are trying to identify that what are the other things that we have failed to identify, please use your machine learning algorithm, apply on our data and give us some results that we have not observed. This thing is also possible in today's era. That is why the final report what you are going to submit, it should have these kinds of details. Also, the test and control articles identified by the name, abstract, code, strength, purity, composition and all the other various details. In my previous lecture you have seen where I have mentioned that when you prepare the protocol, basically whatever the things you actually prepare in protocol, same thing, not only same thing, in a more elaborate way you may prepare your report in a more elaborate way. So, the final report, it should consisting the test and control article, including the name, chemical abstract number or code. You are aware why code is important. I have told you in my previous that is a lecture. Then also the strength, also the purity and the composition. Then the stability of the test and control article under condition of administration. You see, some of the drug molecules we actually do not tell, we actually do not prescribe in summer condition, because at high temperature there are chances that it might be showing some issues because of the higher various enzymatic or hormonogenic activities. So, that is why it should be also there that under which condition it should be administered, including the time when it should be taken, also a description of the method that is actually used. Then a description of the dosage regime and route of administration and duration. What kind of dose you are actually going to give, tablet dosage form, capsule dosage form, or it is a solid, semi solid or liquid dosage form, what kind of dosage form you are going to give, dose regime, how long, and route of administration, by which route you are going to give, oral route or your that is other kinds of intravenous, subcutaneous, or what kind of route you are giving, and the duration, for how long, and a description of the all circumstances that may be affected the quality and integrity of the data. Okay. So, description, in description you will be mentioning that what are the circumstances may impact the quality and integrity of the data. Circumstances like temperature, pressure, storage, humidity. Okay. These are the various circumstances, or there might be many other circumstances. I have just mentioned only few. Okay. So, in description all these circumstances should be there, so that you can able to understand that how it is actually affecting the quality and integrity of the data. If I tell you more, the name of the study director, very important, the name of the other scientists who are working professionals, and the name of the supervisory personnel involved in the study, their name should be also involved. In case you failed in this project, you have not received good sufficient data to that is proclaim that yes, our drug is working on cancer, at that scenario those people will be called. In some cases there is some leakage of from your lab, and it might be creating some health issue, environmental issue for common people. You can call these people. These people will be called to explain, to give the proper reply, and if they have done any wrong work or any illegal work by their own, they will also get punished in some of the cases. You have seen the story of Wuhan Institute of Virology. You are aware that why government of China, also WHO and many of the international organizations, they are getting pressure to Chinese government, to the authorities of Wuhan Institute of Virology also, that if any mistake may happen for the leakage from SARS-CoV-2 from that lab, that should be identified. Things are going on, we have to wait and watch. One of the report has published by WHO, it is a very comprehensive report. No conclusion was given with saying that yes, it was leaked from this lab, but ultimately for future purposes, whether the things are actually associated or not, for that we need to mention the name of the scientist. Also, if you are doing some great work, they will be getting some Nobel Prize. That is why also it is required. A description of the transformation, calculation, operational perform in the data, summary and analysis the data, and a statement of conclusion drawn from the analysis, that thing is also required. And what are the other things? A signed dated report to each of the individual scientist and other professional involved in the study, the location where this specimen, raw data and other final report are stored, and the statement prepared and signed by the quality assurance unit. You see, ultimately the quality assurance people are actually coming here in this picture, because whatever we are actually trying to do, our main work is to ensure the quality, right. So, ultimately the quality assurance people will give you the assurance that yes, it was done in a correct manner, there is no any compromise with the safety issue, and everything was worked in a very perfect manner. That is why quality assurance things will be come to the picture. Then another thing I like to mention you, there will be a statement, it will be given by the quality assurance people. They will not only they certify, they will also tell you the various kinds of protocol, and there are also tell you about various kinds of precautions whatever they have taken. Okay. So, that is why it is very much important. Now, let me tell you more. So, ultimately whatever the final report, this final report will be signed by the study director, because study director he will be observing, he will be analyzing all the various aspects, whatever any mistakes was happen or not. So, after analyzing everything, the study director will give you ultimately the sign, and with this with his sign it will be regarded as yes, it is completed, now we may ready to submit. Now, after submission, if any mistake or any kinds of mishap may they observe, then again it will be evaluated. Then the in point B, after submission of the by the study director, correction or addition to the final report shall be in the form of an amendment by the study director. In case of any correction is required, then in that case the final report or it should be something should be added. The amendment shall be clearly identified by the part of the final report that is being added to corrected, and the reasons of the correction of the addition shall be signed and dated by the person responsible. Also, after submission, in some of the cases the director actually try to that is amend or try to change some reports, because in some of the cases it is actually necessary. Now, after submission, after completion of the all this work, what is next? Next is the storage and retrieval of the record data. Ultimately, data is important. Whatever we are doing, whatever we are working, we are working only for the data. So, the raw data, documentation, protocol, final reports, specimens generated as a result of the non-clinical laboratory study shall be retained. There shall be archives for orderly storage and excipient retrieval of the raw data, documentation, protocol, specimen, intern and the final reports. An individual shall be identified for responsible for the archives. Any only authorized person shall be entered in the archive. See, archive is very important. Why we are actually talking about the archives? By going to the archive, you will be able to see or you will be able to analyze the success and failure of a study, the cost and expenditure of a study by comparing the past and present, and you can predict the future. In future, how much cost it will take or how much time it will take or whether it will be successful or not. That is why we are actually focusing on statistical methodologies or also in modern days machine learning. Okay. So, the material retained or referred in the archive shall be indexed to the permit expended to the retrieval. And also after storing after retrieval of data, so how you will retain the data? Retention of data is very essential. So, record retention requirement set forth in the section 2, documentation record, raw data, specimen pertaining to the non-clinical laboratory studies and required to be made by this part shall be retained in the archive, for which following will be shortest. For example, a period of at least 2 years following of the date on which an application of the research or marketing permitted, in the support of which the results of the non-clinical laboratory study were submitted, reported to the FDA. At least for 2 years, the entire data should be retained, because if FDA is asking that show us your data, any organization is asking, if you are trying to sell the drug in India, you are trying to get the approval of the drug in India, you will have to take the you have to show the data to CDSCO. You have to show the data to CDSCO. That is why it is important. Also, the wet specimens, samples, test, control article, especially prepared material which are related to the fragile and differ markedly in stability and quality during the storage shall be retained only as long as the quality of the preparation afford to the evaluation. Okay. And also the master schedule sheet, copies, protocols, records of quality assurance inspection shall be maintained by the quality assurance unit as an easily accessible system of record for the period of time specified. Okay. And if I tell the fifth, the another point, the summaries of training and experience and job description required to maintain and maybe written along with the other testing facilities employed records for the length time of the specified. Records and reports of the maintained and calibration and inspection equipped shall be retained. You see how seriously we are actually working. Calibration record, okay, inspection of the equipment, every data we are actually preserving, so that in future any super inspection may happen, that time we can counter that thing with our data, that yes, we have done proper calibration. After that we have started our studies. Ground required by this part may be retained either as original records as or true copies such as photocopies, microfilm, microfiche or other accurate reproduction of the original records. All the things should be remain. So, after doing all such things, if a facility conducting non-clinical testing goes out of business, all the raw data, then your documentation and other materials should be transferred to the archive of the sponsor of the study. The FDA shall be notified writing in such kind of transfer. You have to notify the FDA that yes, our work is actually going out of the business. For example, you have started your work with a anti-cancer project, but now you are shifting your entire work for a new disease, for a new kinds of work. In this scenario, you will have to inform the FDA, because ultimately FDA is the authority. After doing your entire work, FDA will give you the approval. So, you will have to tell the FDA. Now, some of the issues in which cases there are chances of the disqualification. What are the purpose of disqualification of the entire study? To permit the exclusion of the consideration of the completed studies that were conducted by a testing facility. To exclude from consideration of the studies completed after the date of disqualification until the facility can satisfy the commissioner that it will conducted studies in the compliance with the such regulations. The determination that a non-clinical laboratory study may not be considered in the support of the application for a research in the market permit does not exist. Now, what are the grounds? Testing facilities fail to comply one or more of the regulation of the part, very important. Many of the cases the non-compliances are adversely affected to the validity of the non-clinical laboratory studies, and also other that is a lesser regulatory actions, warning or rejection, or have not been or will probably not been adequately archive compliance with the GLP. Okay. In those cases it might be get rejected. Now, if I tell you the notice and opportunity for hearing on proposal of the disqualification, whenever your commissioner has information indicating that the grounds of the opinion justify the disqualification, a testing facility he may issue the testing facilities written in notice. Okay. and a hearing of the disqualification shall be conducted in accordance with the requirement of the regulatory hearing. And after that, ultimately you will be getting some final order of disqualification. Whenever the commissioner is informed, indicate the grounds of his opportunity of your this disqualification, he might be giving you the final order. So, that is it from my side. Today we have actually learned the reporting of non-clinical laboratory results, and we have also seen the storage and retrieval of the data. We also have learned the retention of the records, the disqualification of the testing, and ground of disqualification, and notice, and ultimately the final order. So, after getting the final order, everything will be over, then there will be no chance for again to recall or requalification. So, thank you all of you, thank you for being with us in our all the previous lectures. When I have started the unit 3, we have told you a lots of things. We have started with the quality assurance of the packaging material. I have started with the primary, secondary packaging, primary packaging, all those things, how you will ensure that your packaging was done perfectly, how you will ensure the packaging material is correct. From there, I have told you lots of things about GLP. I have told you GLP in terms of animal testing, clinical trials, and all the various aspects. So, thank you all of you for joining us. We have learned together, we have learned a lot of things. We shall continue in our learning in next lectures. I hope some other teacher will conduct the unit 4. So, you may keep learning. So, thank you all of you for being with us. God bless.